7,000 proteins and counting. The ability to measure all of these proteins at once begins with a multifactorial approach, leading to exquisite specificity. But how does exquisite specificity actually enable scientists and researchers to measure 7,000 proteins at once? Let's take a look. As we know, much of biology is about interactions between complementary shapes. This is especially true for proteins, which come in a variety of shapes and sizes. To measure proteins with affinity reagents, we therefore need a large library of complementary shapes. Many researchers use libraries of polyclonal antibodies for selecting affinity reagents, but these antibodies often show poor specificity to their targets. A new class of reagents, called SOMAmer reagents, offers many distinct advantages over standard polyclonal antibodies, including the ability to measure more proteins more specifically. SOMAmer reagents are custom-made to target each protein of interest. They have modified side chains that enhance shape complementarity with their target proteins and don't have the lot-to-lot variability inherent in antibodies. Each SOMAmer reagent is selected from an enormous library of candidate sequences, 1,000,000 billion molecules, until a perfect, exquisite shape match is found for the protein target. Although specificity is now exquisite, in many cases, we continue to add multiple SOMAmers per protein to enhance this even further. Antibody-based assays, which require 2 polyclonal antibodies to approach this degree of specificity, demand more reagents and may create cross-reactivity issues that do not affect SOMAmer reagents. But that's not where it ends. SOMAmer reagents are not only selected for their perfect binding, but also for their slow off rates. This ensures that the correct protein remains preferentially bound to its unique SOMAmer reagent during competition downstream. SomaLogic has developed a groundbreaking technology known as the SomaScan Assay that uses SOMAmer reagents to allow you to measure more than 7,000 proteins at once, each with a high degree of specificity. The SomaScan Assay is a 2-step or 2-catch process that uses a universal polyanionic competitor to ensure high specificity. SOMAmer reagents are made of DNA, which has a negatively charged backbone, and are therefore all polyanionic. This allows the use of a universal polyanionic competitor to prevent rebinding after rapid dissociation, further enhancing target specificity. Use of a universal competitor isn't as effective with antibody-based assays, where any given competitor can have a vastly different affinity to various antibodies. To recap, it's important to remember that the SomaScan assay has a high degree of specificity due to a number of cooperative mechanisms. First, SOMAmer reagents are chemically synthesized to have modified side chains that enhance shape complementarity with their target proteins without needing a second affinity reagent to boost specificity. Second, each reagent is matched to its target proteins based on both perfect binding shape and slow off rates. And third, the 2-catch SomaScan assay design uses a universal competitor that further minimizes nonspecific protein binding. The definitive question that remains is, why take a chance on the underperforming technology of antibody assays? Trust the one that delivers exquisite specificity. Trust the SomaScan assay.